Okay, so why is DNA replication important? Well, you might know that all cells come from pre-existing cells. So if we talk about eukaryotic cells undergoing mitosis or prokaryotic cells undergoing binary fission, we are saying we have one cell that is dividing to become two. And in our starting cell, let's say it's a eukaryotic human cell, we might have 46 chromosomes. We need to make a copy of those so that each new cell gets 46 chromosomes as well. So we actually need to replicate the DNA so that when we divide into two new cells, we have a complete set of chromosomes for each cell. So how does that happen? Well, let's look at the structure of DNA again. So here we've got our double-stranded DNA molecule. It would be a twisted helix regularly, but I've just flattened it out for simplicity. We have these one strand going from 3' to 5', where you can see the orientation of the ribose sugar is, like a little crown here, whereas on this strand, the little crown's on that side. And we have our phosphate, our sugar, and our base, and each one of those is a nucleotide. So what we're going to have to do is create another copy of this. Okay. Now in a human genome, that might be a few billion bases long. I think it's about 7 billion bases long that we have to make a copy of. So how do we do that? Well, we're going to use some enzymes. So the two enzymes that we're going to use to start with initially are an enzyme called helicase and another enzyme called DNA polymerase. So this process is actually quite similar to transcription that we've learnt about already, and that used RNA polymerase. So we've got to make sure that we're using the correct terminology when we're talking about each process. So DNA polymerase for DNA replication and RNA polymerase for RNA synthesis or transcription. So let's move those into position. So now we've got helicase in position and DNA polymerase behind it. Now, in actual fact, it's quite a complex process. I'm simplifying it a little bit here, but for our intensive purposes, this is all we really need to know. So helicase starts at an origin of replication point, and from there it moves along the 3' to 5' strand. So we call that a leading strand. Now either of these two sides could be the leading strand. It will just determine which side the polymerase and helicase start and in which direction they run. But we're starting them on the left, so they can run from 3' to 5', because as we know, we always build new nucleotides from 5' to 3'. Okay, so you'll see that happen in a sec. So as you can see, it moves along, and the DNA polymerase has moved along one, and we've got a free nucleotide doing complementary base pairing with the thymine, and we've got an adenine nucleotide joining with it. Now, the helicase and DNA polymerase would actually move along together, but I wasn't that good with the animation, so I couldn't do that as well as I would have liked. So as DNA polymerase keeps moving along, you see it's quite easy to build from 5' to 3'. 5' here, and then the 3' is on this end. So we can build in that orientation. So the reason why we actually build in that particular orientation is because on that nucleotide, there are actually 3 phosphates, not just one. So we know it's a sugar phosphate backbone, but the nucleotide actually carries 3, and then when it binds to the nucleotide next to it, we actually lose these 2 phosphates and they go off, and that creates that bond next to them. So we'll keep moving along, adding in another nucleotide, and you can see it's quite easy to move along from this 3' to 5' because all we're doing is adding these nucleotides in the 5' to 3' direction. So it's one after another, one after another, one after another. Now can you see a problem? I can, and that's that we have this other strand that's not having any nucleotides added to it. And that's a big problem because to make an exact copy, we need to duplicate both strands. Okay? So as we move along, we're opening up and exposing this area that needs extra nucleotides. So what actually happens is after that area gets exposed, then we come back and deal with that strand. Okay, so we have a leading strand that we can just replicate in one direction continuously, and then a lagging strand that, as you can see, we've exposed some bases as we've moved across. Then we have to go back and do complementary base pairing. Now, it's a little bit more complicated than just adding nucleotides. So after I go through the basics, we'll go back and look at a bit more detail. So we go back and then have to add in those extra nucleotides. Okay, so now that we've done that, you can see that we've now got another problem. When helicase and DNA polymerase runs along there again, you'll see that we can just keep moving our DNA polymerase along the leading strand. But we still have that problem now where we've got now a new set of exposed bases on the lagging strand that we're going to have to deal with. Okay, so I'm just going to draw this from scratch again, just to show you in a bit more detail what's happening. Okay, so let's say we have our 2 strands of DNA. We then use those 2 enzymes to open up a section, and we're going to have helicase enzyme opening up and unwinding that helix, and then behind we would have DNA polymerase matching the nucleotides up to that leading strand. So here's our 3' leading strand to 5', and we're now adding those free nucleotides to that side. On the lagging side, and that would be 5' to 3', on the lagging strand, which is 5' to 3', we then have this exposed section. So this exposed section, and because it will be a section at a time, is called an Okazaki fragment. Okay, so this section here is called an Okazaki fragment, and we've got to deal with that in a very special way. So what actually happens first is we lay down a very short RNA primer. So it's just RNA nucleotides. So they complementary pair with those exposed DNA nucleotides, and so we use an enzyme called primase to lay down a short RNA primer, which is in yellow, and then we get a different DNA polymerase. There's actually 3, I believe, but a different one. For our purposes, it doesn't matter. We have that DNA polymerase from that point on, then move down, and then add in through complementary base pairing the DNA nucleotides, and then we come back and replace these RNA primer with DNA, and then we have another enzyme that moves along called ligase and joins them together. So you can see that process is quite a more complex one than just reading the leading strand. Okay. So the leading strand, helicase opens up and creates this replication fork, unwinding, DNA polymerase runs along and uses complementary base pairing to add a second strand of DNA. Then we expose these Okazaki fragments that we have to then go back, add an RNA primer with primase, then use DNA polymerase to, from that point, go backwards and add bases and create that other strand, and then we go back and replace those RNA nucleotides with DNA nucleotides, and then we use a ligase enzyme to join it all together. Okay, so quite a bit more complicated. But at the end, you'll notice that we'll have, if we look at this top diagram, we'll have this strand and now another strand. Okay, so then eventually during mitosis or binary fission, those 2 strands will then break apart. So if we look at this diagram here, what would that look like if that DNA was to all condense and coil together to form a chromosome? It would look like this. And there's our normal replicated chromosome. Okay? So just remember that this DNA replication process is not happening during mitosis. It's happened well before during a synthesis phase. So I'll just put that up in the corner as a reminder. So that's happening during the synthesis phase of the cell cycle, not during mitosis or not during binary fission in the cell division. So this is happening through the synthesis phase, DNA replication, and then mitosis is actually nuclear division, and then cytokinesis is cell division. So I hope that's nice and clear. And to check whether you understood what I said, if you haven't, ask plenty of questions in the comment section below and I'll get back to you, or even make new videos to explain it differently or in a better way. But hopefully it made sense. If it has, you hopefully should be able to answer this question. Let's say I've got a cell. I'm going to just show the DNA. So there's DNA. And I'm going to replicate that DNA, but instead of using just regular nucleotides, I'm going to use radioactive nucleotides. Okay? So my question would be, what would the DNA look like in the two new cells? So let's say my radioactive nucleotides are red. So we've got the original DNA in black, the radioactive nucleotides in red, so we can see them, they're a different color. So in the next cell, or the next two cells, what would the DNA look like? Let's put it around there. And then if I took this one and replicated it twice, and this time I took away the radioactive nucleotides and added regular nucleotides in again, what would that DNA molecule look like in those two cells? Post your answers below, ask your teacher about that, and hopefully enjoy the video. If you did, check us out on Anytime Education, like that page. For more videos, you can go to anytimeeducation.com. In the subscribe section, tick that, and there should be some videos playing in the left corner of your screen. Check those out as well.